One protein, the HRAS, one protein, and the KRAS codes for two proteins. KRAS for A and KRAS for B. That's for the sake of completion. As a result of differential splicing. Basically, the main difference between these two proteins is that the 4B doesn't have a fatty acid attached to it. Now, what you find these proteins to be expressed differentially in different cells, in different tissues, but when mutations occur in any one of those genes that are associated with a particular class of tumors, they fall into the categories of mutations that we've talked about. They decrease the GTPase activity, and they reside in these two areas of the coding sequence, either ones that bind lead to the site where gap binds, or where the kinetic site resides, the active site. Okay. All right, so now let's just finish up. So when you look at different tumors that involve RAS mutations, and I told you, at least as an operating figure, 25% of spontaneously occurring human cancers involve a RAS mutation. So it's very common. Seems to be the second common type of mutations after the one that is most common, that's P53. In the tumor suppressor gene. But sometimes there are other genetic aberrations in one of the RAS coding genes have been found in tumors. So sometimes you find in tumors that the gene for one of those RAS protein is amplified, so that one of those isoforms of the RAS protein becomes overproduced in the tumor cells. And sometimes what is overproduced could be a mutant form of that protein that's produced at a higher concentration than otherwise, or simply the normal protein that is present at very high, much higher concentration in the transformed cells. So the type of changes, genetic changes that have been linked to these overproductions of the RAS fall into two categories. Most commonly, it is amplification of one of the RAS genes. Not talking about chromosomal rearrangement, that will be the subject of the next lecture, and you will see how chromosomal rearrangements could lead to overproduction of any one protein. But the gene amplification is seen in two forms. So the three different RAS genes are coded on sites at three different chromosomes, chromosome 1, 11, and 12, and we don't need to worry about which of the gene is found on which chromosome. But let us say that on one of these chromosomes, the RAS coding sequence typically would be found, let us say, on the long arm of this particular chromosome. So one form of gene amplification involves the elongation of one of the arms of the homologous chromosomes that code for that form of RAS. And this is detected in karyotype analysis. So you look at the patterns of the chromosomes, and you see that one has an elongated arm. And typically, it's really not known what the intermediate steps are. There are hypotheses, but what you would find is that if this is a site of gene amplification, instead of having on this particular homologous chromosome of that set one copy of the RAS gene, you might find hundreds present in tandem sequences. The other type of amplification is one that's going to be important to talk about later on when we talk about drug resistance. This is double minute chromosomes. I hope you've seen that one before. Double minute chromosomes. So digging back from your genetics, what are double minute chromosomes? Anybody has seen them before? So these look like fragments of chromatin material. They look like chromosomes, except they don't have any centromeres. And they become really visible in a cell that is about to go through mitosis. If I'm going to take a cell here, and let us say that this cell is preparing for mitosis, is going through the process of chromosomal condensation. See the chromosomes, each one of them has a centromere that becomes attached to the spindle fibers. But when you have double minute chromosomes in such cells, what you find are fragments, could be a substantial number, look like almost like particles of dust sprinkled between these condensing chromosomes. They look like chromosomes. They're associated with histones. They have the same structures, except they have no centromeres. And what you find when you see these double minute chromosomes, typically, is that tandem repeats of a particular sequence that becomes obviously amplified. So in some of the amplification of the RAS genes, the tumor cells display double minute chromosomes reflecting a large number of copies of the RAS coding sequence. And as a net result of that, what you find in cells, because you have multiple copies of the gene, you can produce the protein at higher rates, you find that the RAS protein is present at much higher concentration than otherwise. And again, it could be the normal RAS, or it could be the mutant form of the RAS. Both of them are been seen. All right, any questions? So any ideas why, if you were to raise the concentration of the normal RAS protein, that provide the cell with a proliferating advantage? Any ideas? Suggestions? Yeah?
Could it be that you just have more protein serving phosphorylated, and then that can lead to increased transcription factors from phosphorylate?
So let's start out with a cell that has 10 copies of the RAS, and another cell that has 1,000 copies of that wild type RAS. And the question is, why is that cell that has the 1,000 copies, does it have a proliferative advantage, or does it get a proliferative signal over the cell that has 10? And so what happens now in that cell that has 1,000? You said something about phosphorylation?
Yeah, there's increased phosphorylation of the downstream.
So what causes it? So now we are starting out, we're really dealing with unknowns, so there's really no clear-cut answer. So we're just thinking about possibilities. All right, so we have two possibilities. This cell, there is no RAS activation pathway, so there is no SOS1 that can bind to RAS and cause a GDP-GTP exchange. Neither do those cells, actually, because there's no growth factor. So the question has been, could such a cell still have that increased capacity to stimulate downstream? So what would cause that to happen? You may be right, but what would cause it to happen? What we're looking for is a higher concentration of RAS GTP here than RAS GTP there without a growth factor. because that's the only thing that can stimulate RAF to act, right? So what we want is a possibility of having more of this molecule in order for this to become active. So how would an increasing concentration of RAS in a cell cause a higher level of RAS GTP? There's,
Fire gap.
well, but gap is what causes that to be hydrolyzed.
I thought it can pass in both directions.
No, so gap doesn't, SOS does.
Okay.
Okay? All right, so there are lots of arguments, but here is one which is based really on mass action, and it's really not clear whether or not it's correct. So let us say the cell has 10 RAS. So this is a normal cell. Has 10 RAS molecules. And this is the tumor cell, which we won't worry about it now, which has 1,000 RAS molecules. And let us say, let's focus on this. Let us say you need, out of these 10 RAS molecules, to have 7 of them associated with GTP to provide enough of a stimulus to cause the downstream events to take place. Right? So we need 7 of these to be RAS GTP to cause a stimulus. All right, so what happens is that under normal circumstance, the growth factor binds all of this, and now you end up causing more RAS GTP to become produced, and everything happens fine. And then this is turned on. So this is what is needed, 7 RAS GTP. Now let's think about mass action, and in this case, we'll think about the equilibrium. Let us say that this is not quite as irreversible as we have talked about, that this reaction going forward here can go at a very, very slow rate compared to when the growth factor is there. And let us say that at steady state, when there is no growth factor, 2 of those 10 RAS would be in the RAS GTP form. Right? So 20% without a stimulus are RAS GTP forms. Not enough to cause a stimulation because you need 7. Are you with me? Now, if we go to those tumor cells, which have 1,000 RAS molecules, and apply exactly the same mass action, 20% of those at steady state would be RAS GTP, 20% of 1,000 would be 20 RAS GTP molecules would be present without a stimulus. And as a result, this is ample concentration of RAS GTP to stimulate the RAS pathway.
Would it be 200 instead of 20?
I'm sorry, thank you, that's even worse. I should have made this 100. All right. So that's really one of the arguments. All right, any questions on this? But really, whether or not this is the answer is really not clear. Finally, RAS-related sequences. So let's look at something completely different from a mammal, right? We'll look at baker's yeast, Saccharomyces cerevisiae. Right, so these are cells that can grow as haploid or as diploid. They're wonderful cells. And we'll look at cells that can be maintained in the haploid form, right? So they have one copy of each one of their chromosomes. Now, Saccharomyces cerevisiae, when its genome was sequenced and the genome of the gene of the RAS was sequenced, turned out to contain two genes that code for RAS-like proteins. These are referred to as RAS, the protein SC1 for Saccharomyces cerevisiae 1, and RAS SC2. Nomenclature doesn't matter. So there are two genes. They're bigger than the genes than the proteins. Proteins are bigger than what you find in a mammalian cells. They're about 300 amino acids. So one is 302, the other is about 308. Now, you can mutate one of those two genes and the cells will proliferate. You can take your choice. You can either mutate RAS SC1 or RAS SC2 and the cells will proliferate. But if you were to mutate, knock off the function of both, the cells would not be viable. And you can play with this with temperature-sensitive mutations. But for now, for us, let's just say that if the cells are lacking this particular gene, they cannot make the proteins, and you turn off the gene that codes for the other one, the cells will not survive. Now what is amazing is this. If you were to use transfection procedure to insert the human or mouse HRAS coding sequences into the genomes of those cells, you could actually knock off that second RAS coding sequence and the cells would survive. This is a much smaller protein, it's 189 amino acids compared to 302. This is from a mammal, this is from something that makes beer. But the conservation of function is astounding, that you can rescue those cells with a human coding sequence, a mouse coding sequence was tried even with RAS, human RAS from tumors containing one of the 3-point mutations, and it worked in all of these categories. So these proteins play, obviously, very fundamental roles in the proliferation and survival of cells, and so important that their function was conserved to a point where mutations was not really allowed to go too far in changing the properties of those proteins, even over such range of evolutionary scales. Okay. Any questions on this? All right, so now we're finished with the RAS activation pathway, a somewhat shortened presentation. We're going to move into another aspect of oncogene activation, and that's by reciprocal translocation. And actually this is very interesting because these types of aberrations have been associated with substantial number of human tumors, 20% of them. And they can be used, these translocations, as a way of predicting the course of development of a disorder, even before the malignancy manifested itself physically in the individual. As long as the cells are there, one can predict what happens. All right, so we'll go for about 15 minutes, give you a break, and then come back. But before we get started, several of you visited me in the office, and one of you left an umbrella. So the umbrella is in my office. It was raining yesterday. I took it out and I said, oh, wonderful, I forgot it. And then I saw it was a different color, but it's back in my office. Okay.
Today too.
So the topic here, which will be one that will cover over 4 lectures, so this is substantial area, deals with activations of oncogenes by a completely different mechanism than what we have talked about before. And that involves the breakage of segments of chromosomes, 2 non-homologous chromosomes, and the exchange of the pieces from one chromosome to the other, as you will see. And I'll give you two examples. These are the classic examples. So we'll go back to chronic myelogenous leukemia that I introduced in the beginning of the course. But we'll discuss it in greater detail, so we'll look at the stages. But the focus here really will be on the affected loci, the genes that are affected by these translocations, and why the aberrant regulation of those genes can be very potent initiating events in malignant transformation. So look at two genes that are involved in these translocation. One is c-Abl, which I have told you before is tyrosine kinase, and the other one is another kinase called BCR that we'll talk about when we get to it. And then we'll talk about the disease and its management. That those studies on that disorder created a new family of drugs that target specific tyrosine kinases that become active in certain tumors. So this drug appeared in early 2000, and now it is the gold standard for other type of tyrosine kinase inhibitors. That's called Gleevec. And we'll look at another type of malignancy, that's Burkitt's lymphoma. It's a childhood malignancy that you find very prevalent in equatorial Africa. And it is involved, it's also associated with a very specific class of translocation. One of them involves the transcription factors, one of the genes affected, involved, and the other gene that is involved codes for one of the immunoglobulin proteins. And we'll get to it. And then we'll come back to a phenomenon that I mentioned in passing in the beginning, in that in a cell you need multiple mutations in order for the malignant phenotypes to become apparent. So how would you tell whether or not a particular mutation will contribute to the likelihood of the development of a tumor cell if that cell had another type of mutation in it? So this involves development of a new experimental model that we'll talk about, new to this course, not new to biology, and that's the transgenic animals in which some of these oncogenes are expressed. All right, so let's begin. So one of the fundamental traits of malignant cells is that they have an abnormal chromosomal number, or some major abnormalities in the chromosomal pattern. So very typically, more often, much more often than not, tumor cells are aneuploid. They don't have the normal multiple complement of haploid chromosomes. Usually they're closer to being tetraploid than diploid. We mentioned this in cells that become immortalized. We won't deal with this in this section. There very often some deletions are noted where segments of a specific chromosome would be missing in a particular class of tumors. It's always the same segment of the chromosome that is missing. So for example, some tumors of the retina, neuroblastomas, the affected genes were discovered due to the fact that a segment on a particular chromosome, chromosome 13, was always missing. And these deletions really are what led to the discoveries of tumor suppressor genes. Genes whose products, when are not present, contribute to malignant transformations. And we'll come back to this in the next section of the course. Gene amplification, I gave you an example of RAS, but there are many other oncogenes that become overproduced, whose products become overproduced as a result of gene amplification. And we'll talk about MYC here, but later on. And the last one is translocation. And that's really the subject at hand. All right, so translocation means really the exchange of pieces of chromosomes. Either segment is rotated on the same chromosome, put instead of being head to tail, goes tail to tail. The other is what we're going to talk about, exchange between non-homologous chromosomes. So, but before we get started, I want to remind you of how these changes get detected. they get detected by the standard karyotype analysis that had been around for a long, long time, something I did when I was in high school. And much of the discoveries that I'll talk about actually were done with the standard karyotype staining technique. I'll turn on the light in a second. I just want you to see some of the images. So in this process, which we've covered in 3:30, but I'll give you a thumbnail in a second, the chromosomes are stained, and from the staining, you can see some banding pattern on the chromosomes from the location of the centromere and the length of the two arms and the banding pattern. Someone who knows can align those chromosomes and assign them as to which of the chromosomes they are, number 1 through 22, or are they one of the sex chromosomes. So in human, the karyotype for which this karyotype is seen, obviously there are 22 pairs of chromosomes, and then you have the sex chromosomes. The other method, which was developed in the past 12 years or so as a result of the sequences of the human genome, involves a different staining technique, which again I'll cover very briefly, and that's a spectral karyotyping, in which certain fluorescent dyes bind by hybridization to individual chromosomes, and then the chromosomes can be identified not by their pattern, but by the computer. So somebody like me who've never really done this can actually come up with something like this and appear to be very adept at it. Okay. So in order to get these karyotype analyses completed, you need cells whose chromosomes have become condensed. So just to remind you, so if you want to do this karyotype analysis, you really need cells that are proliferating. Because if they're not proliferating, the chromosomes are not going to go through mitosis and condense. That's the first thing. And it doesn't matter where the cells are coming from. There could be cells from a fetus by amniocentesis. There could be cells that are obtained, say, from a bone marrow biopsy by aspiration from one of the bones, the pelvic bones. Those cells have to be maintained in culture in order to proliferate, and then those proliferating cells that enter mitosis are the ones that are used for the stain. So in the procedure, the cells are maintained in culture, then they are incubated with an agent that blocks cells in mitosis, and that agent would be what? So what agent would block cells in mitosis? What disrupts microtubules? An agent. Colchicine. So the cells that are proliferating are incubated with colchicine for about 20 hours or so. And what happens is that cells that were in G1 during the incubation can actually get to mitosis, but because colchicine causes the disruption of microtubules, those cells cannot form the spindle fibers, and then they become arrested at a point in the cell cycle where the chromosomes are condensed but have not been tethered to the spindle. So after that period of incubation with colchicine, the cells basically are dropped on a cover slip or glass slide so that they shatter, and then they're fixed, and then the chromosomes are stained to give the pattern that I've shown you. So at the end of the staining process, what the people who do this karyotype analysis do is that they sit under a microscope and look at individual cells, because those cells would be separated enough. And what they want is a cell in which its individual chromosomes are scattered. So you'd imagine this is a cell, and then this is a microscope slide, there'll be another cell there, there'll be another cell there. And they look at these. So now, first of all, take a look from this very classic paper. So these cells here, for this chromosome, the centromere is here. You can see banding, and you can see the long arm and the short arm. And if you've done this for your career, you can look at this and say, aha, this is chromosome 19. And this might be chromosome number 22. So once I see those and they have been scattered enough, so this actually is not very good because some of these chromosomes here are not spread out, they have so this is not an ideal image. But if it were, what then they do is that they cut these individual chromosomes from the printout. And then they line them up to show you, to give you this figure. So these actually came from these cutout of the individual chromosomes to get this pattern. And you can count how many chromosomes there are, and if you have the eye, you can look at one chromosome and say, aha, this just does not look normal. None of us here have that eye, but I'll show you some example. But one way is, actually, if you look at this, to show you just how little you can see, this actually is an abnormal one. Right? There's a difference here in the darkness of this band, but there's also slightly shorter, and you will see in a moment. All right, so this is the classic technique. And that's one that has been used to identify many of these chromosomal aberrations associated with tumors and arteries. Very laborious and quite required quite an education. Now, the more recent techniques involves the staining of the chromosomes. So you imagine every step I told you is the same way, but the chromosomes are stained as a result of the generation of sequences that would hybridize selectively to particular chromosomes. So if we look at chromosome 1, and because the DNA sequence of that chromosome is known, you could imagine synthesizing, which is what happens, short nucleotides that collectively will hybridize along the segments of that chromosome. And these short nucleotide segments are attached to particular dyes that fluoresce certain color under fluorescent microscope. And the same thing would be done for chromosome 13, different sequence of oligonucleotides. These short nucleotides are generated artificially, and then they are linked to another fluorescent dye. So you have this mixture of dyes that are used to stain the chromosomes. So if you imagine pouring those combination of dyes on those chromosomes, those that will recognize chromosome 1 will hybridize to it, those that recognize chromosome 9 will hybridize to it, and then you have a computer along with a microscope that is equipped with the right filters, can detect the different colors and align them for you. And what you can see here is that there's a problem here with chromosome 18. Chromosome 18 should fluoresce as purple. There is one of the two chromosomes has a segment here that looks like it had come from an X chromosome. So that's obviously some sort of translocation, which is difficult to detect by the standard method. So, from our discussion as we go on, this really has not been applied to those studies. So most of it will be here, and I'd like you to appreciate what it would take in order to identify some of the translocations we'll talk about. Any questions? Let's see what comes next. All right, so before the break, we'll give you this. So one of the way of designating particular regions on chromosomes, and I'm going to be referring to that system, is has been worked out by geneticists. So for example, if we go to chromosome number 8, this is chromosome number 8, and as you remember, when they are stained, the chromosome has a short arm and has a long arm. So the short arm is referred to as P, the long arm is referred to as Q. And then you have some bands. So if there is a segment here of interest, this can be referred to, in this particular case, as 8q, if this happens to be band 24, 8q24. So this designates a specific spot on a chromosome. If it's chromosome 13 and the short arm, it would be 13p, whatever is the band. Right? So, straightforward. And with this, let's take 5 minutes, and then we'll get to the topic. As you can see on the screen. So I'll tell you a little bit about the disorder before we get to the molecular biology, the mechanism. So, first of all, in general, these are chromosomal translocation, and we're looking at reciprocal translocations in which two pieces of two different chromosomes, let's say it's chromosome 8 and 14. Pick two at random here, 8 and 14. So in the reciprocal translocation, and I'm going to make 14 look like this, a piece from chromosome 14 would break, and a piece from chromosome 8 would break, and those two pieces would switch to give the following structure. So I'm just going to put break here and a break there to give a chromosome number 8, which has a piece that came from chromosome 14, and vice versa, chromosome 14 with a segment that came from chromosome 8. And the problem that results from such structure is obviously you have two coding sequences that were previously non-contiguous, not sitting next to each other, are brought into contiguity at the site of ligation. So you can imagine a sequence that existed on this chromosome could be regulating a sequence that had existed on the other chromosome. So these turned out to be very common. About 20% of spontaneously occurring human cancers have these sort of translocations associated with them. They are mostly hematopoietic cancers, cancers that arise from bone marrow cells, and some childhood sarcomas. And the number of genes that have been linked to these chromosomal translocation is substantial. There are at least 360 different genes that have been identified as becoming deregulated as a result of these translocations and associated tightly with certain cancers. So for example, in the case of Burkitt's lymphoma that we're going to talk about, in 100% of the cases, every case, there is a translocation that involves chromosome number 8. And if you were to look at every malignant cell in every child in equatorial Africa that comes down with Burkitt's lymphoma, you find a translocation that involves chromosome number 8. So that kind of tight association has been used now as a way of screening for malignancies in stages long before the tumors have reached point of detection by previously standard methods. All right. So two pieces from two non-homologous chromosomes are exchanged. Certain rearrangements are tightly associated with specific malignancies. And the result in all of these cases lead to overexpression and activations of one of the genes that is involved in the process. And these are typically proto-oncogenes. So for chronic myeloid leukemia now, the translocations that are involved deal with loci on chromosome number 9 and 22. So you can imagine in one of the cells in the bone marrow, because this is really where it starts, such sporadic problem that would have occurred led to the breakage of part of chromosome 9 and the breakage in part of chromosome 22, and then the repair process, which really is triggered when there are broken ends of chromosomes suddenly are present, led to the ligation of those two pieces, and it happened to be that the pieces were ligated incorrectly. So what you find in chronic myeloid leukemia, and that was established in 1960 by two investigators in the University of Pennsylvania Medical School, is that in every case that they examined of chronic myeloid leukemia, there was a shortening in chromosome 22, and that came to be referred to as the Philadelphia chromosome, the location where the discovery was made. So that became a way of predicting the progression of a disease that otherwise, as I said, would not have manifested itself fully. Now you have to imagine how such a link had to be made. So you have these two investigators sitting in hospital. They see patients with chronic myeloid leukemia. And in each case, in order to identify that such chromosomal translocation took place, they had to do a karyotype analysis. So they take some of the cells from the bone marrow of those patients, grow them in culture, stain them, as I told you. But then the problem is the following. They were to say that if you see this translocation, that individual has chronic myeloid leukemia, imagine all of the controls that they had to do in order to link the Philadelphia chromosome, in particular, with that form of malignancy. The controls had to be enormous. And other forms of leukemias also had to be eliminated. So they had to look at patients that had other types of malignancies that did not have the Philadelphia chromosomes, as well as normal patients, and enough of a number in order to establish the connection. All right, so this disorder in the United States affects about 5,000 individuals a year. Slightly more common among men than women. And as I said, it starts out in bone marrow stem cells. But it's detected in its earlier stages as an elevation in some of the leukocytes in the circulation of a patient. So what happens is that during a routine visit, if this disease had not progressed to a point where it can create symptoms, and we'll talk about the symptoms, during a routine visit, a blood sample shows that there is a slight elevation or significant elevation in white blood cells. That could be any one of a variety of benign issues. A person could be dealing with an infection, it could be an allergic reaction. But if a second visit still shows that those granulocytes, those leukocytes are elevated, then a potential problem could be suspected. And what happens then is that the person goes for a biopsy. So there's an aspiration that's taken out of the pelvis typically. And the cells that are removed then are stained by the procedures we've talked about, and the chromosomal pattern is looked at. And if there is that translocation, where are we? That translocation that is detected in the bone marrow aspirate cells, then the prognosis can be made. Early on, if this patient just at the beginning of the stage, you can say without any treatment, next 5 years, the number of white blood cells would continue to rise, but no organ failure, but other side effects are seen that we're going to talk about. And then about 1.5 years after those 5 years, the person goes through an acute phase where the number of white blood cells rise enormously and the person dies of multiple organ failures, typically kidney, as well as respiratory tract infections. But they have other problems along the way. Anemias, for example, bleeding disorders. So this is how the disorder progresses. It's typically for individuals 50 years or older. About 25% of the cases are for individuals over 60 years of age. And in the United States, there's an estimate that there are 20,000 such cases. So it's really not very common, but it's the most common form of leukemias. So, about the cells that are involved. You've seen this slide before, but let's walk through it because I want to emphasize the cell type. So this is a standard blood smear, something that's never done now because machines do it. But basically shows that if you take a drop of cell and you spread it on a glass slide and then you stain it from this standard method, you see obviously the red blood cells are all the same size, no nuclei are the majority of the cells, and then occasionally you see some cells that have nuclei. And those cells that are nucleated, the minority of them, the rarer ones, fall into the categories of lymphocytes. They have particular size. The immature lymphocytes have large nucleus, small cytoplasm. The nucleus is rounded. And then as cells begin to secrete antibodies, some of those lymphocytes enlarge. So this is a larger one, this is a small one. And the enlarged ones have granular material that contains secretory substances, happen to be the antibodies. There are some cells that have phagocytic activities, these monocytes, and then the cells that are in question. So these are the leukocytes, and all of them have one trait in that their nuclei are lobular. That's why they're referred to sometimes as polymorph or polymorphonuclear leukocytes, but that we just deal with leukocytes here. And they have been differentiated on the basis of the staining of their cytoplasm. Cytoplasm has plenty of granules. Now I'm going to turn on the light. You can write. And depending on the staining of the granules, they've been labeled neutrophils when the granules don't absorb much stain. Eosinophils, or eosin, which is a red stain that is found in that subclass of leukocytes. And basophils, another subclass of those leukocytes, their granule stain purple. So these eosinophils and basophils are involved in allergic reactions as well as reactions that deal with localized infection. So they contain substances such as histamine and heparin. So heparin causes the blood to remain thin at the site of the infection. The histamine obviously increases the permeability of the blood vessels. And some of them release agents that cause other leukocytes to proliferate and come to that site. The neutrophils are the most numerous, and these are phagocytic activities, so they digest crud that they run into, but they also, how is that for a scientific term? Debris, it's more accurate, and they have oxidative products that they release to kill potential pathogens. All right. So in the earliest stages of those types of leukemias, one of those subtypes of cells will be found elevated, higher than normal, moderately. And moderately, you'll be surprised at anywhere from 10 to 20 times normal. That, to me, is not moderate, but moderate compared to what happens as the disease progresses. Sometimes the platelets numbers are also elevated, but that does not last. All right. So where are the events that lead to this happening? So you've seen such slide before, some of it in this course early on and other courses. So this is the pathway of differentiations of the derivative of the hematopoietic stem cell. So you have these cells that can differentiate in any one of different subtypes found in the bone marrow, typically the flat bones, the skull, the ribs, pelvis. and some long bone, especially in the femur. So these cells are really very small in number relative to the other cells that are found in the bone marrow. Very, very few, but they survive, luckily. And these different cells can be identified, by the way, as a result of very laborious process that linked certain markers on their surfaces to some of the stages of their differentiation. So nowadays, with fluorescent cell sorters, you can actually sort those individual cells by looking at the staining of certain markers on their surfaces. So the hematopoietic cells, you can think of them as dividing, where one of their progenies remain as a stem cell, and then another one of their progenies then would lead to some of the derivatives of that stem cell, depending on which growth factor exists. If there's plenty of erythropoietin, you would imagine that this pathway would be stimulated. If you have granulocyte colony stimulating factors, you don't need to worry about that, but some factor that would cause these myeloid cells to differentiate towards the leukocytic pathway. All right, so the products are the granular leukocytes that we're interested in. Some of them are the megakaryocytes, where fragments of their cytoplasm are generated to make the platelets. And of course, products that we're not interested in, red blood cells, as well as lymphocytes. So you have a population of these myeloid dividing cells in the bone marrow, in the process of generating these terminally differentiated cells that eventually you see in the circulation. So the problems that we're going to talk about reside in a pool of some of these myeloid cells. So you could think in the bone marrow as one of those translocations that we've talked about having occurred in one of those cells, providing the proliferative advantage that we're going to talk about in great detail. The number of cells with that translocation then would begin to multiply. And then over time, those cells become more and more numerous, and that is reflected in the amount of products that they secrete. So what happens with these myeloid cells, and why is the translocation so effective? So in the case of the translocation, as you will see as we develop this, it has three effects, these 9 and 22 translocation. It generates a product that is a tyrosine kinase that stimulates those cells to divide. Basically, stimulate the RAS activation pathway. So that's one effect. The other effect that this product has is anti-apoptotic. It prevents cells that have that modified product from going into apoptosis. So those cells survive. And the third, it blocks the ability of those cells to repair double-stranded DNA breaks. So you end up having cells that have these chromosomal aberrations that basically over years accumulate, and eventually you have a cell that is very, very aggressive. Not only does it have the translocation, but has other mutation that cause some of their progenies to be produced at very, very high rate. And as I said, from beginning to end, that process can take anywhere from 5 to 7 years without any intervention. Right. So as almost a closing slide, let's see what comes next. Yeah, so this is the closing slide. So if you were to look at a patient who has progressed further than these earlier stages, to the point where you begin to see some of those malignant cells spilling into the cytoplasm, the products of the malignant cells. So this would be a blood smear, and there are some cells in here that look normal. So this looks to me like one of those small lymphocytes, right? This could be a monocyte, could be a malignant cells. This could be a normal leukocyte. You can see the nucleus is distinct, and the cytoplasm has neutrophil-like particles, but this cell doesn't look anything at all like what you should find in the blood. This one, not. Right? And at high magnification, you can see cells like these don't look at all normal. This may or may not be normal. You can't tell. It's within, the play. This could be normal, but who knows? But clearly, this is not normal. This is not normal. The nucleus is really swollen. This is not normal. It has 2 nuclei, etc. But by that stage, the disorder would have spread for a few years. So where we're going to go then with this discussion is first, we'll look at some of the symptoms of the disorder, and then we'll look at the loci that are involved, what are the products of the genes that are involved do in order to promote this proliferation, and then finally, intervention method. What drugs are available that can help patients deal with this? And next slide. Okay.